The fatty acids that are saturated fats, monounsaturated fats, or polyunsaturated fats, with the structure of what's called a fatty acid starts with this functional group. So a functional group, remember, is a bunch of atoms that are bonded together and they have a single function, kind of like a polyatomic ion. They don't separate, they work, and they're grouping of atoms. So this functional group is the carboxylic acid, COOH. I can see that I just got the message that I have an unstable internet. Let's cross my fingers that I'm not going to crash. But remember, if I crash, I'm coming back. So the carboxylic acid function group, starting point for the fatty acid structure. So let's start with what is a saturated fat. So saturated means that all of the carbons and hydrogens have saturated fat. And so let's put 18 carbons on this. What you can see is that these are the line, this is the line angle drawing. And in a line angle drawing, every end of the line, like right here, and every joint where two lines meet and there's a bend, like here, or even here where there's more bonds in there. End of a line is a carbon. And so there's no letter C there. And there's also the number of hydrogens is implied so that every carbon has 4 bonds coming out of it. So that means that this carbon at the end has to have 4 bonds. So there's 1 going to a carbon, and then there must be 3 on there. Or any of these bonds that is any of these carbons that is a joint. So that's a bend, a bend, a bend. Each one of these joints is a carbon that has 2 bonds to other carbons. And so there's 2 hydrogens on there also. And this compares to this right here at this joint. There's 1, 2, 3, 4 bonds coming out. So that means that carbon doesn't have any extra hydrogens. So when I put this in with the atoms, that's the structural formula, that carbon at the end, carbons at the joints where 2 lines meet has 2 hydrogens, and then that carbon group doesn't have any extra hydrogens. Okay. Now, the saturated fat means that all of those carbons are bonded together with single bonds. There's no double bonds in there. And saturated means that these bonds are saturated with the maximum amount of hydrogens. That's what a saturated fat is. It is saturated with hydrogens, which means there are no double bonds. Okay. Once you add in a double bond, you have less than the maximum number of hydrogens. And so that's why it's called unsaturated when you add in a double bond. For the monounsaturated, or polyunsaturated is more than 1 double bond. Space filling model, a straight line. And line, when you actually fill this in with the atom, that's a 1 thick straight line. So this is a saturated fat. And so what saturated fats do is they stack up, right? And that's why saturated fats are solids at room temperature, the coconut oil or butter. And it's also why saturated fats are unhealthy fats, is that they will stack up on the insides of your arteries and lead to plaques and blood, particularly in the cardiac artery, that would lead to a heart attack. So this is the saturated fat. Even though when you look at it like this line, zigzag, once you fill in all those atoms, it's an overall straight structure and can stack up. Let's take a look at the monounsaturated fat. So again, you start with the functional group, which is called carboxylic acid, carbons on. So those are the same 18 carbons as a line angle drawing, but you can see here, this is a double bond that is in the cis arrangement. An arrangement where both of the hydrogens are on the same side. So the same side. So that's this causing the rest of the carbon chain to bend around. So that's a bent shape. And the things that are the same though in this line angle drawing is the ends of the lines, the ends of the line, and anywhere where 2 lines meet, a joint, are here, and you can count 18 of these. Okay, there's a carbon here too. And then the hydrogens are implied. And so the structural formula shows us that the carbon at the end is a CH3. All of those inside are CH2s, except for that double bond. So remember, it's called unsaturated because there's less than the maximum number of hydrogens when you introduce a double bond. So that double bond has an extra bond there. And then instead of CH2, that's just C and 1H. Instead of CH2, that's just C and 1H. So that's why it is no longer saturated with the maximum amount of hydrogens when you introduce a double bond. Okay, so this is an overall bent shape, right? And it's pretty different from the saturated fatty acid. So unsaturated fatty acid, which has one double bond, is called monounsaturated and has a bent shape. So what happens when you go to a polyunsaturated fatty acid? So polyunsaturated fatty acid is when you have more than one double bond. You can have 2, 3. And so here's 18 carbons with 2 double bonds. We're going to start with the line angle drawing. So these are 2 double bonds, and they both are the cis arrangement. So the cis arrangement forces a bent shape. It forces the carbon chain to bend around because the 2 hydrogens are on the same side of the double bond. And so here's the structural formula. You can see that each carbon in the double bond has less than the maximum number of hydrogens. 2. That's why it's called unsaturated. And then we have the space filling model, which shows us a really bent shape. Okay, so what the bent shape and the really bent shape have in common is that they don't stack up. So when you have the polyunsaturated fat that has the cis double bond, those don't stack up. And the cis fats and polyunsaturated fats, those are healthy fats because they don't stack up. They flow. And that's what I showed you what olive oil is. It's a liquid at room temperature. And then there's the essential fats that are in foods that your body needs that you can get from, say, flaxseed, avocado, salmon. These are polyunsaturated fats that humans can't make, but you need them. So you need to eat them from food. That's why they're called unsaturated fats. Okay, so let's take a look at the worksheet. So I'm going to switch over to that and we can fill in some of this information. This is from last lecture's worksheet. This is the second page of the worksheet last time. So this was in the 10.1 lecture worksheets, but we're working on it today. So this one we started last time, the 10.1 lecture worksheet, the first page of fats and oils. So that was a little review about saturated, monounsaturated fats. I did want to bring up again that the trans monounsaturated fatty acid, that's the unhealthy one because the arrangement of the double bond is not bent, and that gives an overall straight shape. So we looked at that last time as how trans fats are usually indicated on a nutrition facts label because they're as unhealthy as saturated fats because of this. So now let's continue on. So this is the beginning of today's lecture. So this is 11.1 lecture, and this is the second page of the lecture worksheets. So this is a polyunsaturated fatty acid. It has more than one double bond. There's 2. Here's 1, 2. Both of those are the cis arrangement. That cis arrangement is that when you have the double bond is bent. It's a bent shape. Okay, so that's because both of the hydrogens in the double bond are on the same side, forcing the carbon chain bent shape. So there's an overall bent shape of the entire molecule. And this does affect the ability. It does not form a solid, it forms a liquid because it can't stack up. These molecules cannot stack up. Okay, it's, it could be really arrange these perfectly to stack up. The straight shapes can easily stack up, okay? And so this polyunsaturated fat is not considered an unhealthy fat. It's a healthy fat, okay? In fact, 2 saturated fats are essential fats, and essential fats is the human body needs these. but you can't produce them. So you have to eat them. And these two polyunsaturated fats are called linoleic acid and also linolenic acid. And so we'll look at a breakdown of the foods that have linoleic and linolenic acid. Are superfoods that I refer to, like salmon and avocado and flaxseed. And so if you've heard of omega-369, I mentioned that in the video, omega-369 is the essential fat. It's also has 3 double bonds, 3, 6, and 9. That's why it's called omega-3, 6, and 9. The double bonds on that fatty acid chain. Okay, so when you are considering what a nutrition facts label, here's an example. Bags of chips. So chips usually have tons of fat in them, and not all of them are unhealthy fats. So the unhealthy fats are the saturated fats, saturated fat and trans fat. Those are the unhealthy ones. And that's why they are indicated on the nutrition facts label. And the monounsaturated and polyunsaturated are also indicated, but really for people on fat restricted diets, those are the 2 fats they're looking at specifically. Okay. And so when you have the amounts of fats, they're given as mass, but we can do calculations to calculate the percentages of the fat so that it's not all unhealthy fats. Some fats are healthy fats. Okay, so how to calculate the percentage when you have a total? So first you need your total fat, which is 10 grams. So when you're doing a percentage, just like when you calculate a grade percentage, you need the portion. So the amount of saturated fat is 1.5. The portion divided by the total, 10 grams, and then times 100%. So when you have the portion divided by the total, the units are the same, grams and grams. And so the units you'll end up with are percent. And so that's 1.5 divided by 10 and times 100%. Saturated fat. Okay. And so that is not this. Oh, that's percent of daily value. That's different, right? So you can't use that like an answer key. The trans fat is 0 grams. So that would be 0 grams over 10 grams times 100%. And so this is grams and grams will cancel out. And then this don't need a calculator for is 0 trans fat. Okay. So there's also some polyunsaturated fat and monounsaturated fat in there. So the polyunsaturated fat has 6 grams. So this is going to be 6 grams divided by the same total. Remember, I'm using the same number every time, which is this 10 grams. So this divided by 10 grams times 100%. And so the grams and grams will cancel out. And the units you're left with will be percent. And so you have 60% of the polyunsaturated fats in the total. So that's not bad. It's not all unhealthy fats. And then the monounsaturated fat is 2.5 grams out of the 10 grams times 100%. And grams will cancel out. No, don't do that. Do this. Okay. And then this will be calculating out to 25%. Add up the total percentages, that's going to add up to 15 plus 60 plus 25. It should add up to 100%. Okay, so that's what's going on in this label is they're trying to tell you the unhealthy fats, and that can help quite a bit for those on a fat restricted diet. So I know that these molecules are more complicated. At the same time, they're also more simple because they're just carbon, hydrogen, oxygen. So they're just more atoms bonded together. And this on the screen is a triglyceride. So the triglyceride is the glycerol, and you want glycerol and then find the 3 fatty acids. So the easiest way to look for the glycerol is to look for a whole bunch of oxygens, because glycerol has 3 OH groups, and those fatty acids bond through those 3 OH groups. So looking for oxygens is really the easiest way to go about this. So I see a whole bunch of oxygens in here. Okay, so that's the glycerol molecule right there. It doesn't have OH groups because there are 3 fatty acids bonded through the OH groups. And so now they're just oxygens. Okay, so there's 1, 2, 3 carbons, and there are 1, 2, 3 oxygens. And then what's coming off of those 3 oxygens are 3 fatty acids. Okay, and so let's click on this, and this will bust this out to its component parts. So here's the glycerol molecule. This glycerol molecule is 3 carbons, it's 3 OH groups. Now there are 3 fatty acids, and so we can see these are 3 different fatty acids, and that's why foods can have components, which are that graph that I showed you, that box graph, which like olive oil is saturated fat, some polyunsaturated fat, and some saturated fat, because olive oil is made out of triglycerides that have all these fats. These are the fats. These fatty acids are the fats that are in foods. So this is an example here of the saturated fat, not this is a saturated fatty acid. And how do I know it's saturated? Because it's all single bonds on there. Okay. This over here is an example of a monounsaturated fatty acid. And how did I know that? Because there's 1 double bond right there. And then this is an example of the polyunsaturated fatty. And how do I know that? Because there's 1, 2, 3 double bonds there. Okay. This is the glycerol molecule, and that glycerol molecule stands out once you've separated all of them. Okay, so let's take a look at the, there's one more I want to look at, which is this one. Okay, so actually let me make this bigger. Okay, so let's say you have a butter blend like this. I'll slow this down. If you've had a butter blend, it's typically margarine, but butter is a natural animal fat, but this is a blend. And what butter blend is, it's basically trying to replicate butter. It's a spreadable solid. And clicking on this, it's made out of triglycerides. So triglyceride molecules, that's these complicated looking structures, and these are the line angle drawings. And so what you want to look for when you are given a triglyceride is start by looking for the glycerol. So that is going to be looking for the bunch of oxygens. And so here is the glycerol is the center of this. Okay, and here's another one with the glycerol is the center with those oxygens. Each of these triglycerides has 3 fatty acids coming off of it. So let's start with this one. So this is these has 3 saturated fatty acids, and they are all single bonds, and they are all straight. Okay. This triglyceride down here has a monounsaturated fatty acid and also a polyunsaturated fatty acid. So let's start with this one. Okay. So this is a saturated fatty acid, and so in this butter blend, there's monounsaturated fatty acids, and then there's polyunsaturated fatty acids. So all of those fatty acids are present because they are all parts of the triglyceride that are all in this butter blend. Okay. And so these are given, this is where you get those grams on the nutrition facts label of the saturated fat, the monounsaturated and polyunsaturated, is going back to the original fatty acids. Okay. So if we want to figure out the percentage, remember you take the, let me get my calculator again. You take the portion and divide it by the total to get the percentage. So for the saturated fatty acid, this is 6.4 grams, and the total is 12. 8. So I'm going to take 6.4 divided by 12.8 times 100%, and that's a 50%. Okay. So you could do this for the saturated, the monounsaturated, polyunsaturated, and other to get all the percentages. Sorry. And that adds up to 100, just like we did on the worksheet. So with those percentages, we can actually use this to identify what this is, what this butter blend is. So what is the unknown triglyceride? It has to match this percentage of 50% saturated fat, 42% monounsaturated, 4% polyunsaturated, and 4% other. So going by the colors, 50% saturated is looking for 50% of the blue. So what goes to 50% on the beef tallow is 50% here of the saturated fat. And then let's see, there's also about 42%. Yep, that goes from 50 to 92 of the monounsaturated, the yellow. And then you have the polyunsaturated and the other. So that is that triglyceride in there, is this identified by taking the percentages and matching them on this identification of the various types of fats and oils in foods. So you will see this in your Quizlet assignment for today's lecture, that you'll see the same type of identification process. So the process of hydrogenation gives these fatty acids some different properties, the properties that are more desirable physically, in that they're no longer liquid, but they're more of a solid. So that's a semi-solid. And also, in theory, no longer, less unhealthy or less healthy, but also having the properties of being semi-solid is a good thing. So you kind of have a mixture of healthy, unhealthy when you start off with a healthy fat like oleic acid that has one double bond, and turn it into less than one double bond, or a mixture. Some of them are still double bonds, and then others are single bonds. So that's called partial hydrogenation. So soybean oil, which is a healthy fat, is less healthy, but also having properties that are more semi-solid and spreadable, as opposed to an oil that's just a liquid. So that's what the process of hydrogenation, adding hydrogens across a double bond. So here you have a monounsaturated fatty acid. This is called oleic acid. It's in oil. So it's why olive oil can flow. It's a liquid at room temperature, because it has that bent shape. And so in hydrogenation, it takes some of the molecules, not all of them, but some of them, and adds hydrogen across the double bond. See what happens here. And instead of having the double bond, now with the process of hydrogenation, you have a single bond. And so this will end up with not all of these double bonds become single bonds, but some of them. So you'll have mixture. So you'll have some, which is partial hydrogenation. You won't have all oleic acid, you won't have all stearic acid, but you'll have a percentage of each in your end result of the partial hydrogenated fat. Okay. And so this ends up with a mixture of these, which are liquid, these which are solid. And so that's how you have a semi-solid fatty acid. And so you'll also end up with a mixture of this being healthy and this being unhealthy, a mixture of kind of halfway in between healthy and unhealthy. But you have the properties of both, which is kind of what sometimes consumers want. Okay. With carbohydrates, these are the second major group of macronutrients. So the carbohydrates are the sugars, starches, and fiber in foods. Now we'll go back and look at a nutrition facts label, but let's first look at the structures of these carbohydrates. So the name is carbohydrate, and what that means is hydrated carbon, or carbon plus water. So each carbon, every single carbon, has a water molecule, and a water molecule, which is H2O as the chemical formula, you could really write it as a hydrogen and a hydroxide, H and OH. So in a carbohydrate structure, each carbon has a hydrogen bonded to it, and a hydroxide bonded to it. So on either side of this carbon that I just highlighted in blue, there's hydrogen and hydroxide. That's one water molecule that's bonded to that carbon. Okay, so it's not bonded as H2O. First, you split the water molecule, and then you bond the hydrogen and hydroxide on either side of the carbon. Now, you're not just going to have one carbon, but you'll have a series of carbons. So there can be another carbon above, another carbon below. You're going to usually have 5 or 6 total in your common monosaccharides. And those 5 or 6 carbons can be straight chain, but more commonly, we're going to be looking at rings. Okay, so carbohydrates start off as the simplest version as monomers. That's called a monosaccharide. Saccharide called glucose. So this is glucose, which is a 6-member ring structure. So 6-member, that means 6 points. And it's called 6-member because these points are not all the same atom. So each one of these is the line angle drawing here that shows a hexagon shape. But remember, when you have the line angle drawing, there are no carbons or hydrogens shown as the atoms C or H. So where you have 2 lines intersect here, that means that's a carbon. When you have 2 lines intersect here, that's a carbon. Here's another carbon. Here's another carbon. There's 3 lines intersecting right here. And those 3 lines intersecting is another carbon. So in this hexagon shape, there are 5 carbons plus 1 oxygen. That oxygen that is the 6th member of that ring. So that's why it's called a 6-member ring. It's not all carbons, it's not all oxygens, but there are 6 total atoms in there. This is 6 carbons total because there's a carbon above it in this CH2OH group. So there are 6 carbons total, but the ring itself in glucose has 5 carbons plus 1 oxygen. So there's 2 versions of glucose, that monosaccharide that's a 6-member ring. There's this version and this other version. Right. So first, let's make sure it's pretty similar. There's 1, 2, 3, 4, 5 carbons in the ring plus an oxygen. So it's still a 6-member ring. There's 1 carbon outside of the ring. So it's still 6 carbons total. Okay. It's a 6-member ring structure. And the only difference between these is the position of 1 hydroxide group, one of those OH groups. So in alpha glucose, the OH group on carbon number 1 is pointing down. So let's first show the numbering so we know which is C1, which is carbon number 1. Okay, so that's to the right of the ring oxygen. Okay, so the ring oxygen is this, highlighting in red, and to the right of that is this joint, which is carbon number 1. So I'll write carbon number 1, C1. Now this one is carbon number 1. And so the numbering of these carbons is the number increases going in a clockwise direction. So this is carbon number 2 right here. And then this is carbon number 3 right here at that joint. This is carbon number 4. This one right here is carbon number 5. And then this right here above the ring is carbon number 6. So those are your 6 carbons total. But this carbon number 1 has that hydroxide group on there, that OH. And notice that OH is pointing down. So that's in purple, that OH group is down. All the other OH groups are exactly the same in the other glucose version, which is called beta glucose. So beta glucose, we first want to find carbon number 1. And in that carbon number 1, it has that OH group pointing up. So first, let's find carbon number 1. Start by finding the ring oxygen that's highlighted in of that ring oxygen is that carbon, which is that joint. So that's carbon number 1 right there. And then number the carbons increasing going around in a clockwise direction. So this is carbon number 2, carbon number 3, carbon number 4. This one is carbon number 5. And then above this outside in CH2OH is carbon number 6. So there's 6 carbons total. And that carbon number 1 for the beta glucose has that OH group pointing up. So that's the only difference, is this OH group is pointing up, which means it's above the ring. So the ring is a flat ring, like a disc. So picture that like a Frisbee. And then you have an OH group above the Frisbee. And then this compares to the alpha glucose, which has the OH group down below the Frisbee, that disc. Okay, so this has to be considered in 3 dimensions. And so that's really the difference, is between beta glucose is the only difference is that one OH group. You can see all the other OH groups are exactly the same. Carbon number 2 is down, carbon number 3 is up, carbon number 4 is down. And so the only difference between alpha glucose and beta glucose is the OH group on carbon number 1. So alpha glucose has the OH down compared to the beta glucose that has the OH up. Okay, and so you might be thinking, oh, why does this matter? Because it makes the difference in humans' ability to digest these bonds when you put these monosaccharides together. Okay, so remember the difference between alpha glucose and beta glucose, or refer back to this. Let's take a look at the fructose. So fructose is another example of a monosaccharide, except for it's a 5-member ring. So 5-member ring looks like a pentagon, and just like the 6-member ring, not all of the atoms are carbons. So let's start with this oxygen that's part of the ring, and then to the right of it, this is a carbon. Then here's another one, here's another one, and here's another one. So there's 4 carbons as part of the ring, but then there's also 2 carbons outside of the ring. So there's one here and there's one there. So there's 6 carbons total. Okay, just like with glucose, there were 6 carbons total, but only 4 of the carbons are part of the ring. So in numbering the carbons, the numbering of the carbons is still the same. So you start with carbon number 1 is the first carbon to the right of the ring oxygen. Okay, so the ring oxygen is that oxygen right there that I just highlighted in red. And so carbon number 1 is to the right of that. So that's carbon number 1. However, I got to start actually with this carbon that's outside of it. So that's number 1 is just outside of it. So that's technically the first one to the right is the one that's to the far right, I guess, outside of the ring. So that's carbon number 2. Here's number 3. Continuing on, the carbon numbering increasing as the going in the clockwise direction. Here's carbon 4. Here's carbon number 5 at that joint. And then this going outside the ring is carbon number 6. Okay, so numbering those carbons, I started at the right side of the ring, but outside of the ring this time. This next is moving down. Looking at when you start to put these monosaccharides together, you'll have a disaccharide when you put 2 monosaccharides together. So sucrose is table sugar. Table sugar is called sucrose, and it is a disaccharide. That is 2 monomers put together. So this is 2 monosaccharides bonded together. Okay, so the 2 monosaccharides are glucose and fructose, or the 2 structures that we just looked at. So here you have, this is the glucose is the 6-member ring. That's this one. And then the fructose is the 5-member ring. And that's this one. So glucose is typically the sugar or the saccharide, the monosaccharide that circulates in your blood, and it's blood sugar. it's in a lot of foods. And when food is broken down, it's broken down into the sugar parts. And this is the most common currency of sugar or monosaccharide in your blood. Fructose is fruit sugar. It's the most commonly found in fruits and broken down, and your body can digest both glucose and fructose. These are bonded together. With this is a bond that is called an alpha, or an alpha. This is the symbol alpha, the Greek symbol alpha, bond. And this is a bond, or I'm going to call it a linkage more commonly. So it's an alpha linkage. I'll write that word out, alpha linkage. And notice that alpha linkage has this shape. It has a V shape. This is a bent shape. That will become important when you put together more than one monosaccharide. When you start to put together lots and lots of monomers, when you get a polysaccharide, how they're bonded together will become very important, okay? And so this is the bent shape. There's a single bond between those two monomers, the glucose and the fructose, and it's the bent shape, the alpha linkage. So what humans can do, humans can digest that alpha linkage, because there's basically an enzyme in the human body that can break down that alpha linkage. Probably that bent shape is part of the importance of it, because we'll see that proteins that are enzymes recognize molecules, not because they can't see, they don't have any brains, but they have molecule shape. So when the humans can digest that alpha linkage, that will then release the glucose and fructose. And once they're released, humans can digest those. So digest those individual glucose and fructose monosaccharides. And what I mean by digesting is you can break those bonds, and when you break all those bonds, you get energy from those bonds. So it's going to become important in understanding that humans can digest the alpha linkage, which connects these two sugars. And that's how sugars produce energy and give us 4 calories per gram, is from breaking the bond side of a carbohydrate molecule. And so this starts with, when you have table sugar, you first have to break these apart, and then you have to break all those bonds apart, and that gives you energy that's measured in calories. Okay. Take energy from the sun, and they create molecules that they can use. So when you are eating a potato or corn or sugar, it's originally coming from plants. That's all plant-based food, natural foods, not processed. And the plant didn't make the corn or the sugar. Storing their own sugars. And so, yes, we do take advantage of the fruits of plants, but plants are very smart. They take the energy from the sun and store it in the bonds of carbohydrates that are stored in polysaccharides that are starches. Plants also take the energy from the sun and store it in the bonds still as glucoses, but then take those same glucoses and linking them together create their own structural support. So plants make their own food, they make their own, houses, I guess is to think of it, their own support system. And you got to appreciate plants just from the one molecule glucose. So let's take a look at these polysaccharide structures. So a polysaccharide, as the name implies, is a polymer of polymers that are monosaccharides. So the monosaccharides are those individual single ring structures. So you can have hundreds of those monosaccharides bonded together. And we're looking at these 2 examples of starch, which is in the food that we can eat. that humans can digest. And the reason why we eat starch is because we can digest it. And what we are digesting is the linkage, the bonds between those glucoses. So humans can digest glucose. That's not an issue. Okay. What humans can digest is also the linkages between those glucoses. So we can digest those alpha linkages. And then when you digest this alpha linkage, that's that bent shape, it frees up those glucoses, and then you can break all those bonds in the glucose, just like with the sucrose molecule, the disaccharide. So that's good. So that's good. That's why we eat starch. But remember, another thing I mentioned in the video is that this alpha linkage leads to an overall structure of the monomer that is a coiled. And here's why. So let's write one glucose is, I'm going to draw, instead of this 6-member ring structure, I'm going to make it a black circle. Okay. And so glucose like this is going to be a black circle. So if I'm going to fill this in, that would be 2 of those. But remember, we can have thousands of these. So that's 1 glucose. So I'm going to do a whole bunch more of these. So here's 1 glucose, and it's linked to the next glucose with an alpha linkage. Let's do that in red, just like that. So the alpha linkage is a bent shape. It's a V shape. So that's 1 alpha linkage linked to a glucose. Okay, so let's connect another glucose with an alpha linkage that has to be a bent shape like that. So this bent shape, the next glucose, ends up not next to it or not in a line, but it's going to actually curve around. So here's another glucose. I'm going to link to this with an alpha. This next glucose will start to curve around. And so I'm going to put one more here. Another alpha linkage, which is this bent shape. And oops, no, not that, this one. You can see that what's to happen is that this is going to coil around, and this is the first coiling around. It'll have an overall, okay? So that's an overall coiled structure. And this does relate to its function of energy, because you can store hundreds or thousands, many of these glucoses many of those glucose monomers in a compact coiled shape. Okay, so basically when you roll something up as a coil, that's one of the best ways to compact a bunch of stuff. So when you roll something, like if you've ever packed your clothes in a luggage, when you roll up your clothes, that's really a great way to compactly store things, as opposed to, you know, other methods. And so this is a great way to store energy. That's the goal, is to store energy in those glucose. So if the goal is to store energy and compactly, then this overall structure of the coiled structure is the best way to do that. Okay. So cellulose, though, is also a whole bunch of glucoses. But what's different is the linkage between those glucoses. It's the beta linkage. And humans cannot digest those. So that's the interesting thing, is we can't break down this. And so these glucoses all stay stuck together. Okay. So even though humans could digest this, even though you could digest all the bonds in that, you cannot break those apart. Place. So you can't access any of those glucoses because you can't break apart the glucoses in the first place. So for that reason, cellulose can't provide any energy, because you cannot break apart those glucoses. And so they're all still stuck together. That's why it's called something different. It's dietary fiber. is the other term for cellulose, and it is indicated on a nutrition facts label as part of carbohydrates, but not giving any calories. Okay, so there are no calories, you will notice, from dietary fiber. So fiber does have an important role because it can basically, when you eat food that has starch, you'll have the starch and that provides calories. When you eat foods that has fiber, the fiber goes through and basically kind of cleans out the internal digestive system as it works its way through, because it is not digested. Okay, but here's sketching the overall structure is also important. So again, one glucose molecule, I'm going to draw as a black circle. Okay, so this is one glucose is going to, and instead of linking them with the edge, is an overall straight shape. So it's straight, but it's on the diagonal. That's what that linkage is, compared to the alpha linkage that is that bent or V shape. Okay, so let's have one glucose linked to another glucose, linked to another glucose, linked to another glucose. See where I'm going with this is that these are all linked on the diagonal through that linkage. And what happens is you get an overall straight line, right? It doesn't matter, it's on a diagonal, it's a straight line. And so this is an overall straight shape. And it is a good way then bundle these up. Okay, so you can bundle up. They're basically a bunch of straight lines, and these can be very strong when you bundle these up. And so that's why the fibers in plants, in grass, in stems, in wood, those provide structural support. Gravity. And that's also part of the goal of a plant, is a plant wants to get more close to the sun because it gets the energy from sunlight. And so growing upwards against gravity, well, plants figured out a way to do that by putting these glucose molecules together in a way that they can now structure as opposed to storing energy. Okay, and so this is the overall goal of plants is met so perfectly with just this one molecule of glucose. Just the difference is how those glucoses are bonded, either the alpha linkage or the beta linkage. Okay, so let's